In this video, we will discuss how this aspirin can help in suppressing of the metastasis by limiting the interactions of TXA2 and T cells. In the previous video, we have understood that when this TXA2 interacts with the T cells, it will prevent the activation of the T cells, and which indirectly leads to the increased metastasis within the cancer affected patients. So, however, in this video we will understand how this aspirin will prevent the metastasis by limiting the TXA2 and T cells interactions. Thereby, the T cells activations will not be suppressed, and it can help in preventing the metastasis of the cancer cells. So, how they come up to this aspirin is that the inhibitors of the TXA2, that is COX enzymes, are known to be affected by this aspirin. So the hypothesis that aspirin might also plays a role towards this limiting this TXA2 and T cells interactions. So what it is, they did the in vivo studies where they gave the mice with water this aspirin, and one mice do not give any aspirins. Aspirin experiment is on the panel B. First we will discuss about panel A. In the panel A, they gave mice in water with this TXA2 analog, and one mice does not get any TXA2 analog, and what they are able to find out is that this TXA2 analog mice, and which is affected with this intravenously injected with the pancreatic cancer cells, has more metastatic nodules compared to the vehicle case, in which there is no TXA2 analog has been provided to the mice. And when they use the and when they check this TXA2 interactions with the T cells using the stable metabolite, that is TXB2, they are able to see that the TXB2 are high in the case of vehicle compared to the case where aspirin is there, indicating that so this TXB2 will form when this TXA2 interact with these T cells, and those TXA2 interact with the T cells, forming in the stable TXB2 is found high in the vehicle case but not in the aspirin case, indicating that this aspirin might preventing the interactions of TXA2 and T cells. And when they check the metastatic node in the ARFGF1 present within the cells and ARFGF1 is knocked down, they are able to find out that in the case of ARFGF1 is knocked down, they are able to see decrease between the vehicle and the aspirin. But in the case where ARFGF1 is already knocked down, they do not see any changes, indicating that it's the ARFGF1 that plays a role towards the suppression of this metastasis, that plays a role towards the suppression of this activation of the T cells. And when the ARFGF1 is present but it is not been activated because the TXA2 and T cells interactions are been limited by the aspirin, they are able to see reduced metastasis. But in the case where ARFGF1 is already not present, they do not see too much changes. And same things they have validated using the lung cells, where they check the phosphorylation at S6 positions, and they are able to see that this phosphorylation at the S6 positions is in the CD44 plus CD8 plus T cells is high for the control with vehicle compared to the case case with this is high for this control with aspirin, and ARFGF1 knockout with aspirin and control with aspirin, and also control knockout with vehicle. Because in all these three cases we have seen that in the knockout cases, because ARFGF1 is not present, that leads to the high phosphorylation conditions, and in the control cases where aspirin is present, we can see the high signals of this phosphorylation SC, because the TXA2 and T cells interactions is limited, and that will prevent that will prevent the inhibitions of this phosphorylation at S6 and thus limited the metastasis. And the relationship between phosphorylation and S6 we have discussed that in our previous video. You can refer to that video to understand the relationship of this phosphorylation at S6 and this CD44 plus CD8 plus T cells. Thereafter, they wanted to check the impact on this ARF-GF1 knockout and the control vehicles with aspirin and with TXA2 analog and aspirin both. And what they are able to see that in the case of ARF-GF1 knockout, they do not see any changes within the tumor weight formations, but in the control case, they are able to see less tumor formations. Less tumor formations in the aspirin case and also in the aspirin case, but same results are not visible for this ARF-GF1 knockout, indicating that is further substantiate their hypothesis that is the ARF-GF1 TXA2 cells, the TXA2 ligands interact with the T cells, and through this ARF-GF1 signaling pathways will suppress the activations of the T cells, and that will cause high metastasis. Whereas in the aspirin, in the control case, we see a low metastasis compared to the vehicle. And they validate these things on the right hand side of the panel, where we can see that in the vehicle, the metastatic node is high, but with aspirin, this metastatic node goes down. P-value is around 0.03. But when they check about the ARF-GF1 knockout, we do not see any significant changes, as P-value is greater than 0.09 for all the three cases. So this validate their pathways that they have find out that TXA2 interact with the T cells, and then through this ARF-GF1 will prevent the activation of the T cells. And then they check the metastatic node with this TP to find out the role of TP in this metastasis suppression or of the metastasis, or whether then they use this TP inhibitor to find out the role whether TP plays to find out the role of the TP in the metastasis of these cancer cells. And what they are able to see is that when they use the TP in the wild type conditions and with aspirin, they use aspirin, they see a reduced metastasis. But when they use this TPXA2R knockout, or when the TP inhibitor is used, the changes are between the vehicle and aspirin is non-significant, indicating that is the TP pathways also plays a role along with the ARF-GF1, as this TP signaling pathways is the downstream of this ARF-GF1 that we have discussed in the previous videos, and that also plays a role towards the suppression of the metastasis. And that also plays a role towards the metastasis of the cancer cells, and its inhibitions or knockout will cause suppression of the metastasis. So I hope you understood how this aspirin plays a role towards the suppressions of the metastasis by limiting the interactions of TXA2 and T cells. We have not covered all the figures. Some of the figures we will cover in our next videos, and those figures are not shown here. We have covered all the figures that are all the explanations of the figures that are shown in this slide. We will cover some of the more figures of this research paper in our next videos. That's all for this video. Thank you for your time.